Hello, everyone. So welcome to the Cells, Tissues, Organs, Young Investigator Roundtable discussion. My name is Ryan Gilbert. I'm a professor of biomedical engineering at Rensselaer and one of the three editors-in-chief of Cells, Tissues, Organs, specifically in charge of the tissue engineering section. I'm joined with the other editors-in-chief, Gujun Cheng and Christoph Wiebon. Gujun, if you could introduce yourself, and then we'll go to Christoph.
Sure, yeah. Thanks, yeah. Thanks, thanks all for joining this nice Zoom discussion. So my name is Gojun Shan. I'm based in Japan, Kumamoto University, and I'm a professor and focused on developmental morphogenesis. I got training mainly in developmental biology, and now because of the stage of developmental biology that I'm working on has something to do with stem cell biology. So it's kind of at the crossroads between developmental biology and stem cell biology, and that's what I'm promoting as well for the tissue, cells, tissues, and organs. Christoph, you can.
Yes, hello. My name is Christoph Wiebon, as Ryan said, and I'm a medical doctor, trained medical doctor, but turned into anatomist some time ago. But I worked in clinical medicine as a surgeon and had some clinical training at the Middlesex Hospital in London. Otherwise, I'm been based in Germany all the time, and I'm trying to represent development and been working on the development of early mammalian embryos during the time of gastrulation, and got to know the epithelium mesenchymal transition during that time and during that process. And I took over the CTO together with Ryan and Gudrun in 2018, and we tried to combine the tissue engineering, stem cell biology, and I try to represent the link to clinical medicine, and just also represent the history of that journal, which started off as Acta Anatomica, heavily based on anatomists and medical research. I'm very much looking forward to this session. And I'm very grateful that you can all join and spend some of your precious time. Thank you.
Okay, thank you very much, Gujun and Christoph. We also have some of our editorial board here, so if we could have them briefly introduce themselves. We'll start with Dr. Sethu.
Hello, I'm Palaniappan Sethu. I just go by Sethu. I'm at the University of Alabama at Birmingham. I'm a biomedical engineer by training. My lab really focuses on tissue chip models. So we're a tissue engineering lab. So we build cardiac tissue, vascular tissue, and renal tissue.
Okay, great. Thanks for joining. All right, Dr. Snowetz, if you could go, please.
Yes, my name is Fred Sinowatz. I am Professor Emeritus from the University of Munich. I studied both veterinary medicine and human medicine, and I spent about 8 years in human anatomy, and later I went to veterinary anatomy, histology, and embryology. My interests focuses on embryology and the early development of bovine embryos, and also during the last few years on angiogenesis in the corpus luteum, which is a quite good model for angiogenesis.
Okay, great. Thank you for joining. Really appreciate it. Dr. Zudema, if you could go next, please.
Sure. So my name is Jonathan Zudema. I'm a Marie Curie Fellow in Italy currently. So I'm at the Mario Negri Institute for Pharmacological Research, and my research focuses mainly on nanotechnology combined with biomaterials towards different repair, mostly nervous system repair.
Great. Thank you, John. I think we have somebody else from the editorial board.
Kat, can you introduce yourself?
Yeah, if you could go ahead.
Apologies for arriving late on the call. So my name's Kat Tadjantanakis. I'm a faculty member, and actually I chair the developmental biology program at the Sloan Kettering Institute, which is actually part of Memorial Sloan Kettering Cancer Center. My research interests pertain cell fate, tissue morphogenesis, and early patterning events in mammals using the mouse as an in vivo model, and then mouse and human pluripotent stem cell models as sort of avatars for sort of early development. And my expertise are in genetic engineered mouse models, high resolution imaging, and single cell genomics.
Okay, great. Thanks, Kat, for joining. Appreciate it. All right, Nick, I see that you joined. Would you like to introduce yourself?
Yeah, sure. Can you hear me?
Yep.
Yeah, so I'm joining late. I was dropping my kids off at a camp. Anyways, my name is Nick Leipzig. I'm at the University of Akron. I work in tissue regeneration, mostly in the spinal cord, using bioconjugate techniques and stem cell delivery.
Okay, great. Welcome, Nick. Okay, so I think that's all from the editorial board. So why don't we transition to the awardees? So first of all, congratulations to all the awardees for being selected for this past year's list of young investigators. We're very pleased to have this opportunity for you to share your work. And so what we'll do now is we'll have each of you introduce yourselves. And so why don't we start with Dr. Bao?
Thanks, Ryan, for the nice introduction, and it's really nice to meet all the editor and some of the editor board, and I'm very honored to be selected as one of the Young Investigator Award this year. And as Ryan mentioned, my name is Xiaoping Bao, and I'm current assistant professor in Defense School of Chemical Engineering at Purdue University. And I was trained as a stem cell bioengineering, and I kind of working at the intersection of different biology and kind of engineering medicine using stem cells. After establishing my own lab at Purdue, I kind of turned to make or convince human proven stem cell to become hematopoietic stem cell and immune cells for mainly for translational applications. For example, the cancer immunotherapy. So we kind of refer to make CAR neutrophils for to treat glioblastoma in mouse models. So this kind of my brief background. And again, it's nice to meet you all tonight.
You're great. Thank you very much for joining. We'll go to Dr. Gulbrand. Sarah?
Yeah, hi everyone. My name is Sarah Gulbrand. I'm an assistant professor in the Department of Orthopedic Surgery at the University of Pennsylvania, and I also have a joint appointment at the Philadelphia VA Medical Center. My training is in biomedical engineering, and my lab's research is focused in the area of orthopedics, primarily in studying spinal degeneration. So we study both the pathomechanisms of disease and sort of use that knowledge to come up with new tissue engineering-based approaches to treat spinal degeneration. So it's great to meet everyone and looking forward to the discussion.
Great. Thanks, Sarah. Let's go to Kyle Lampe.
Good morning, everyone. I'm Kyle Lampe. I'm a recently, excuse me, a recently tenured associate professor at the University of Virginia in the chemical engineering department, and I have courtesy appointments in biomedical engineering and neuroscience. Our lab focuses on developing biomaterials for potentially translational applications to deliver cells, as well as creating models of disease or injury. And we focus on neural tissue with a particular emphasis on glial cells, including oligodendrocytes and oligodendrocyte progenitor cells.
Okay, well, welcome, Kyle, and congratulations on being promoted in tenure now. Great news.
Thank you.
You're welcome. Okay, so let's go to Dr. Ozdemir. If you could introduce yourself, please.
Hello, everyone. My name is Tuğba Özdemir. I am an assistant professor in nanoscience and biomedical engineering department at South Dakota School of Mines. And my lab focuses on developing biomaterials to modulate extracellular matrix assembly to control different cellular plastic processes such as EMT or fibroblast to myofibroblast transition. And our focus is to prevent fibrotic capsule formation.
Okay, great. Thank you very much, and congratulations on being selected an awardee. And then we'll finish up by Dr. Sugimara.
Hello, this is Leo from University of Hong Kong. I'm being assistant professor, my third year, originally hematologist, trained as stem cell biologist. We working on like iPSCs, hematopoietic, immune cell differentiations. Exactly, yeah. I mean, I heard from Dr. about that's interesting to discuss. We working on timely antigen receptor macrophages. And actually, about today, yeah, I mean that award, the paper review I wrote was kind of different system, like a new model. Hopefully, like as useful as animal models or new type of organoid to assess functionality of cancer immunotherapy. I'm happy to discuss this. Thanks.
Okay, great. Well, welcome. Okay, so now thank you for all those introductions. Now what we'll do is we'll have a discussion about your submission. So again, as a review for all those that were selected as awardees, each of you had to submit a paper. What we'll do is we'll go in order that we did originally for the introductions. And I'd like for each of you to discuss how your paper fits into your ongoing research, briefly describe the impact of the paper as you see it, and possible next steps with your research, okay? So we'll start with Dr. Bao, and the title of his submission was Adoptive Immunotherapy, a Human Pluripotent Stem Cell Perspective. Dr. Bao?
Yes, thank you again, Ryan, for the introduction. And I think this is the review paper, and we kind of summarize kind of the ongoing challenge associated with the classic CAR T therapy, mainly for cancer immunotherapy. And from a kind of cell manufacturing point of view, we kind of propose to use or leverage the power of human pluripotent stem cell, particularly induced pluripotent stem cell. So we review current or ongoing work from like different stem cell labs around the lab who develop new protocol or new genomic editing tools to make off-the-shelf CAR T cells, CAR natural killer cells, CAR macrophages, and many other like CAR immune cells or hematopoietic stem cell therapy use like human pluripotent stem cell as an original cell source to how to better make these therapeutic cells for target cancer immunotherapy. So this is very, again, brief summary of what we review in that manuscript. But if you're interested, I recommend people to read that paper, or if you have any question or interest, for sure reach out to me, and for sure looking forward to the discussions.
Okay, great. Thank you for that overview. We'll go next to Dr. Goldbrand. The title of her submission was A Tunable Calcium Phosphate Coating to Drive in Vivo Osseointegration of Composite Engineered Tissues.
Awesome. Thanks, Ryan. Yeah, so the background kind of behind this paper is our group over the past several years has been trying to develop and translate a whole tissue engineered intervertebral disc replacement to perhaps eventually one day replace fusion surgery for patients that have disc degeneration and back pain. So the idea is instead of doing a fusion, we actually replace the degenerative disc with a whole tissue engineered living replacement that can hopefully function as a native disc. We've done a lot of work in the past on translating this up in size to match nearly that of a human cervical disc space. and also translation through small animal models to now a large animal model. And one big challenge we see when we're trying to evaluate these and get them to succeed in our large animal model is that integration of the tissue engineered construct with the native bone takes a really long time to develop. So we were interested in modifying the interface between our tissue engineered disc and the native bone by coating it with hydroxyapatite to accelerate the formation of a vascularized mineralized interface. And when we embarked on this journey, we were going back to the literature and using, going back to other folks who have done hydroxyapatite coating for sort of other applications. And we found a lot of kind of inconsistencies in the literature. So the first sort of part of the paper presents really a tunable, robust coating with real validation of the coating on these porous polycaprolactone scaffolds. So we hope that is useful in general for the field for people embarking on trying to hydroxyapatite coat PCL scaffolds. And then we went on to show that it does indeed enhance bone deposition, both in vitro and in vivo, and then also showed that it doesn't do anything detrimental to our tissue engineered discs, at least while it's during in vivo culture. So adjacent to something that should be chondrogenic, it doesn't inherently cause mineralization. So we hope that this is useful for sort of all sorts of composite tissue engineering approaches. And of course, our next steps now that we've validated this are to hopefully apply it in our large animal model studies in the future.
Okay, great. Excellent summary. Thank you, Dr. Gulbrand. Okay, we'll move to Dr. Lampe, and the title of his submission is The Effects of Cell Adhesive Ligand Presentation on Supramolecular Hydrogel Formation and Encapsulated Oligodendrocyte Precursor Cell Growth and Morphology. Kyle?
Yeah, thank you. So our group has been working with different hydrogel types for growing neural cells and trying to understand how different features like integrin binding ligands and stiffness might direct neural stem cell differentiation down different pathways. More recently, we've looked at these oligodendrocyte progenitor cells, which are a lineage restricted progenitor cell, and ultimately give rise to myelinating oligodendrocytes. So that's kind of the direction we were trying to go. We, about 4 years ago, became interested in, to us, an alternative material type. We had previously worked with synthetic based hydrogels, and a grad student got really interested in peptide based materials, these individual small molecules that under the right physiological conditions, controlling pH and salt concentration, spontaneously assemble into nanofiber hydrogels. And so this paper continues our early work with that material. We named it, because it was a brand new sequence, we named it RAPID, or Rapidly Assembling Pentapeptides for Injectable Delivery, right? So the idea being that these could be mixed with cells, they spontaneously form a hydrogel, thereby, kind of agglomerating with the cells. And that material is shear thinning and self-healing. So you could load it into a syringe and it'll shear thin as you apply pressure to the syringe. It will flow through the needle and then very rapidly become a hydrogel again on the other side, right? So this paper explores the idea of adding in integrin binding motifs. So previously we showed we have elegant control over controlling the stiffness, get beautiful nanofiber morphologies, the cells interact with them to some extent. But it's quite popular for good reason to modify our materials, and lots of people try to modify their materials with these integrin binding motifs that are derived from collagen or fibronectin. So we focus on RGD, IKVAV, and YIGSR, which are derived from these kind of naturally protein materials, and said, can we add those onto our small pentapeptide, and will that change anything about the peptide assembly, the mechanics of the material, or their ability of cells to interact with it? And so we did that, and our first exploration was trying to understand peptide assembly using a simulation approach, using molecular dynamics simulations, and simulating these with an atomistic approach and a physics-based model to start to understand how these peptides assemble, and what are the physical interactions, the weak intermolecular forces that give rise to these assembly, and saw no significant impact of these kind of integrin binding ligands on our peptide assembly at physiological concentrations. Saw no change in the stiffness of the resulting hydrogels in a rheology experiment, and saw the start perhaps of some significant changes on cell morphology. So we feel like we've been able to show, we can add these in, get beautiful hydrogels, and have this independent control of stiffness and biochemistry, and thereby impact our oligodendrocyte progenitor cell fate.
Okay, great. Thanks for that summary, Kyle. Let's move on to Dr. Ozdemir. And the title of her submission is Developing Fibrous Biomaterials to Modulate Epithelial to Mesenchymal Transition.
Thank you, Dr. Gilbert. So we submit a review article, and my research on EMT actually developed itself very serendipitously when I was doing my postdoc. I was trying to build a growth factor releasing hydrogel, nanofibrous hydrogel-based material to study branching morphogenesis of a kidney epithelial cell type. And when I was making these core shell fibers, the fiber diameter significantly decreased, and the control fibers were so large diameter, and the growth factor releasing one was small diameter. And we found this epithelial cells forming cluster on the small diameter, and then the large diameter, they were all individual and spindle shape. And then I started looking at these morphologies, and until that time, I didn't know much about epithelial to mesenchymal transition, actually. And then that paper turned out to be an EMT paper, published as an EMT paper. And I think that's how I got into this EMT problem. And later on, I started thinking, usually EMT is associated with cancer metastasis, and in the literature, especially with the biomaterials. And we started thinking not only that, but also there is 3 types of EMT, and each of them is associated with a unique biological process. And, as we summarize in our paper, but in the lab, we are primarily currently focusing on EMT from the wound healing and regeneration aspect. And we are developing fibrous biomaterials using electrospinning. And we recently find that another peptide sequence that is able to attract hyaluronic acid, hyaluron-binding peptide sequences, actually showed interesting abilities on our fibers to form more epithelial-like morphology on the epithelial cells, like keratinocytes, but also on the fibroblast side, it can control myofibroblast to fibroblast transition too. So that's how we are currently working, and we think that this has some implications on not only wound healing, but also foreign body response, especially it's a big problem for implanted biomaterials, implanted tissue engineered constructs. So that's in a nutshell how I end up with this paper.
Yeah, thank you very much for that summary. It's interesting. Sometimes we start research and we end up in a completely different area. So thank you for that. Okay, so we'll finish up by having Dr. Sugimura talk about his submission, which is Give Them Vasculature and Immune Cells, How to Fill the Gap of Organoids.
Yeah, thanks. Yeah, thanks for this opportunity. So that was review, and we were indeed working on that actual paper going to submit. So the idea is you have any organoid, say brain organoid, tumor organoid. Usually they don't have blood vessel, nor immunocells. Exception is you implant in vivo, force vascularize. But what happens is mouse blood vessel, because if we put in mouse brain, that's mouse blood vessel basically penetrate. But you might need human blood vessel for angiography or any of receptor ligand, spatial transcripts analysis, which is routine in my lab. So here idea came, you use iPSCs and make vascular organoid, which is quite easy. And you are already seeing this product on my Zoom background. So this is how our 3D network of blood vessel looks like. 1 millimeter diameter, and you kind of force fuse with, let's say, cancer organoid. And after 4 days, you actually get vascularization. You basically change some medium composition, do some tweak. Now you have immunocells, primary macrophages from iPSCs, because that's quite easy lineage map. Some RBC, red blood cells, and precursor of NK cells. So that's a type of immunocells you can immunize your organoid. And I think that's quite powerful system to understand cancer vessel and immunocell interaction in 3D format in vitro. Yeah, I think that's basically my point. Thanks. Yeah.
Great, thank you very much for that summary and a very nice vasculature in the background there. So thank you for that. Okay, so I worked with all of you in your submissions, and all of you were great to work with, and I think the papers that resulted are all of very high quality. So again, thank you for your submissions. So next, before we open the floor to talk about research, we have a couple of other topics that we wanted to discuss in this roundtable. And the third point is being a young investigator. And so what we would like is for you to discuss opportunities and challenges that you face as young investigators, and maybe focus it in the realm of publishing and how to get your papers out there. So if we could start, and this can be an open discussion, so if any of you would like to go first, we can go from there. So being a young investigator, I guess, Kyle, you're no longer young, right?
Not technically, nope.
But any of you, what are some of the opportunities and challenges? If I could start off, I'm no longer young, right? But many years ago when I was starting out, we didn't have these opportunities to be part of these special issues. And so I think the really nice thing that I've seen over the past, 5 to 10 years is the generation of many journals creating these special issues that highlight young investigators. And so I think it helps young investigators publish and get their work out there. But any of you, what are some of the bottlenecks, some of the challenges in terms of publishing your work?
Yeah, maybe I just made a comment. Yeah. Oh,
Sure.
Oh, sorry. Yeah, thanks. Yeah.
Sure.
So
Yeah,
we were in Hong Kong, Asia, and kind of, from US. So I see big difference of here, Hong Kong and US is we TIs are pretty much, assessed by how many papers we publish, impact factors, like that. Oh, I don't like that. But basically we are like young, weak versus weaker side. So I think to us, new motivation initiative I took part is kind of, activity of e-life does nowadays, more with, bioarchive preprint. If like, I mean, here we don't score any of bioarchive, it's 0. But I'm sure like if the paper is foreign, that's highly recognized in scientist community. And actually, I mean, eLife and this ambassador, I am a member. We were organizing a few activities this year, like having some symposium, virtual, especially now we are focusing this on Asia Pacific region because our metrics is quite specific. We want like open up eyes of our audience in local, society. So yeah, that's basically what we are doing as young PIs.
Yeah, I think that's a great perspective. So bioarchived submissions and being a part of eLife, again, that wasn't possible, within the last decade. So definitely that's a great opportunity. I would agree. Anyone else?
Yeah, Ryan, I would just agree with you on the sort of special issue front. I've noticed that a lot of journals are doing this for young investigators. Sort of within my spine community, we have a journal, JR Spine, that has also a Young Investigator Award, and they have, other special issues that have discounts on publication fees. I think honestly, the biggest hurdle for a lot of us young PIs is a lot of journals are really expensive to publish in, even if they're not open access. So that's hard for me. It's something you need to budget into your startup or your grants and be cognizant of. So I'm also a big proponent of bioarchive and getting things out there early. And luckily for us, at least in the US, the NIH does, seem to support those as, being on grants, and they count them, quote unquote. So I think that's been a really big development in the scientific field of publishing.
Yeah, great. Yeah, I agree, Sarah. It's gotten very expensive, and it seems like journals are going more towards open access and having the investigator pay for all the fees. So the nice thing about cells, tissues, and organs, if I could give a brief plug, is we offer the subscription. So all submissions you don't have to pay for. We do offer the open access privilege, but as all of you are awardees, all of your submissions are open and free. So that's a good thing about this program as well. Anyone else like to share opportunities, challenges?
Yeah, something that Rio said prompted me to think about how to, there's a lot of possible journals that you could submit your work to, right? There's been an impressive proliferation of the number of journals that all could conceivably fit your work. And so, as a young investor, I guess I shouldn't use that anymore, right? This will be my only Young Investigator Award because I won't get any more. But as a former young investigator, I guess the one big challenge for me is trying to figure out where this work fits best, right? When I'm submitting a paper, what journal do we send it to? Maybe I do it wrong in that I kind of write the paper for the most part first and then kind of make a decision about where to send it. I suppose you could kind of target a specific journal as you're writing it. The impact factor game. So I think Rio was talking about, assessment, right? And while we're not supposed to care about impact factor anymore, it very much still plays a role in all of our career trajectories. If for no other reason than, higher impact factor journals tend to have broader readership and more people are likely to see your work, right? And so trying to kind of figure out where it should go, you're always trying to get, as many people to see your work as possible, right? But, if you do that too much, then your paper never gets published because, broader readership expects more data, more citations, more whatever, right? And so that's been a challenge for me, is trying to figure out where to send things appropriately. Thankfully, I guess I've only ever had 2 papers rejected, which maybe means that I'm not aiming high enough with some of the work. I don't know. But I just wanted to put that thought out there, tagging off of something Rio said.
Yeah, no, I think that's a really important point. It's very impressive. You only had 2 rejections, so that's great. You know, from my lab, we always try to aim high in terms of impact factor. But I would agree, one of the challenges is if you don't get into that high impact journal, then you have a paper in that specific format. And then if you want to go to a different publishing house, you have to change the whole format around, and that just delays some things too. I found that one of the things as a mentor, you have to train your students about all the journals that are out there, or the main journals. And so they have to become aware of, you know, where we submit to and what's most appropriate for their work. And so that's a challenge to getting your students kind of up to speed as to where their work might eventually end up. So great perspectives so far. Anyone else would like to share some challenges, opportunities in the realm of publishing?
Yeah, so I kind of just want to maybe add one or more two kind of short point. Nowadays, like this journal, we have kind of this round table discussion. I think nowadays, particularly during pandemic, there are many journals over this kind of similar round table discussion, or just a one-on-one meeting with kind of the decent journals, or like many other journals nowadays. You can very easily to kind of meet the editors. So I think this is a very good opportunity to showcase your like research story or scientific story to the reviewers, to the editors. But I think for me, because I'm working kind of for in the intersection of stem cell biology and kind of engineering medicine. So sometimes if you want to get published in kind of more decent paper, which I kind of now call as a high impact journal, for example, Nature method, or like a cell stem cell, which kind of top 2 journal in my kind of general field. Sometimes you can like with this opportunity, it's easy to go through the editors. Sometimes if your work is good and solid, but still sometimes you will get really mean review comments from the reviewers. Also, like for example, I have a paper submitted to Cell Stem Cell. So the first round, all the reviewers saw that our paper are very like like a very novel, and you need a more kind of technical like a revision, for example, more data, more like more sample per kind of group. But they never give young faculty as a chance to revise your work, to add more. Also, we can rebuttal, but the editor also accept our rebuttal and go to the reviewer. But the second round, the reviewer doesn't like really give you any good comments. They just feel like don't want you to publish. It's not a kind of very good suggestion or comment on improve your story, but just mean like again you are not for again. So this is something kind of really disencourage young faculty, which I think makes me thinking like why like the peer review system maybe need to change, or to make some improvement to kind of more I mean a little favor to young faculty, not just to focus on kind of the senior labs, or kind of the bigger legend in the field. I mean like their work are good, but I think there may be more encouragement can be like, or improvement can be done to encourage publication from young faculties. For example, to make a special case or like issue like a this journal, I think they may like have similar like activity to encourage that.
Yeah, I think it's a great point. So as an editor, and maybe Gujung and Christoph can also comment, as an editor, we are responsible for receiving the critiques or the reviews. and then we have to review what's been stated. And I would agree, sometimes we do get some reviews that say, well, this isn't enough work, or you need to do more work to have it be of appropriate quality. And so I think as an editor, in the editor's role, we have to make sure to kind of navigate through these reviews to make sure that the review is fair. So if you get a critique back and it says you need more work, and that's really all that they state, and I've had those same critiques on my papers that I submit, then I think it's up to the editor to say, well, maybe we can not weigh this critique or this reviewer as much as maybe some of the others that have more constructive comments. But I think it's a really good point. I agree.
Maybe I can just add in a kind of a advantage that I discovered while I was writing the review article for our research was, I thought, it was a lot of people, when they start their independent career, they start with a review article to showcase their ideas. But also, I think it helps you, envision how you are managing your own projects within the lab. So everything go hand in hand almost while we were drafting this. And I think definitely writing a review article, I kind of think that it's a nice strategy to begin in the beginning. This is our one of the first papers as a group. So yeah, I enjoyed it.
No, I think it's a great idea. So I think review papers, so, in terms of, what we published, a lot of emphasis from my department is placed on the research papers, not so much on the review manuscripts. And I would agree with you. I think that especially with a new student, if you can have them write one with you, it really kind of starts them off in the lab knowing the research area quite well. So, several of you submitted review articles, and I thought that they were very well done. And I think that they kind of provided a roadmap as to where you want to go in your work. So I would agree with that. Okay, great. Well, thank you for that very robust discussion and very helpful comments. So before we open it up to general questions, I want you to briefly comment on your future directions. So all of you presented very fine work in your manuscripts. So if you could talk about what your ambitions and thoughts on how you would like to take your research and to continue and develop it. So if we could maybe start off with Dr. Bao, if you could talk about where your research is going in the future.
Yeah, thank you again, Ryan. And my lab kind of after establishing my own lab, I make a little bit change from kind of cardiovascular tissue engineering to more immune engineering using human peripheral stem cells. So in the near future of 5 years, we will continue to develop a new protocol to make CAR engineer based immunotherapy. And one of the focus is as I briefly mentioned, the CAR neutrophil. So because prior neutrophil, they are very short life. So I think at least a good fit to use or leverage human peripheral stem cell. You can genetically engineer the stem cell and then differentiate into all the way from human peripheral stem cell to hematopoietic stem cells, and then to the neutrophil or like a neutral progenitor. So they can bypass the short life of neutrophil. Instead, you can make good cells. So that's a good thing. Means we can have the model to make a human engineer neutrophil. So we want to expand at least kind of platform to study like as a kind of cancer biology, because one of the major challenges in the solid tumor field is kind of immune resistance or the very kind of bad tumor microenvironment. but neutrophil, they can penetrate. So we can see if we can build a model using human stem cell or mouse model in vitro and in vivo to see if we can like integrate the tumor micro environment, particularly the neutrophil, to see if the neutrophil after engineer, they can be better in combination with like a CAR T cell or other immunotherapy to better treat the brain tumor. That's my focus for the next few years.
Yeah, that's a really interesting future direction. I think it's a great idea. Yeah, you're welcome. So Dr. Gulbrand?
Yeah, thanks, Ryan. Yeah, I think the sort of future directions of my group are hopefully translational in nature. I've always been interested in sort of translational work and kind of bridging the gap between like bench and bedside. So perhaps not with our tissue engineering work right away, but with some of our work on studying sort of pathomechanisms of disease. I'm really trying to, over the next couple of years, get to studying this in actual human subjects instead of animal models. Animal models are great because we can control a lot of the variables and really, study things in a less heterogeneous way. But people are inherently heterogeneous, and disease is heterogeneous in people. So one of my big goals is, I've never been trained in clinical research, but that's kind of, I have a career development award through the Department of Veterans Affairs that's wrapping up in the next few years. And kind of the last major goal of that is to kind of bring some of the science into people. We will continue to strive to do that as well with our, tissue engineering or regenerative medicine approaches. With this tissue engineering work in particular, it's a really sort of unique translational conundrum to me, as it's not entirely a cell therapy, it's not entirely a device. It's this very complex tissue engineered structure that I'm pretty sure the FDA will have no idea what to do with it either. So we're working with hopefully finding some industry partners that have experience in sort of large scale cell manufacturing and those sorts of things to sort of guide us towards how we could potentially one day get this in people.
Yeah, that's a great perspective. I think, all of us really want to get our work into human patients, and it's very much a challenge. So it's great that you're working towards that and you have a mechanism with your career development award. That's great. Dr. Lampe?
Yeah, so many ideas about what to do next. Trying to keep this to a minute or so. Following up on Sarah, one thing we're trying to do is take these materials that are part of this current paper and start. We have an animal protocol approved, so we're hoping by the end of the summer to start administering those as neural stem cell delivery vehicles in a rat model of ischemic stroke. So that's an immediate next step, which we hope will have some interesting results by measuring things about the neural stem cell survival and localization as a function of the hydrogel delivery process. And the other aspect that this paper really leaned into a lot is our use of atomistic molecular dynamic simulations to understand things about molecular interactions, which is not something we'd really studied before. We'd done a little bit of molecular dynamic simulations in the past, but not to this amount of detail or with the goal of really getting at mechanism of peptide interactions. And so one both really joyful part of this, as well as a limitation, I suppose, is that work has largely been done by undergraduates in my lab, who are stupendous, but that also requires a lot more input as a PI on really directing and then trying to quality control the work. But it's relatively low risk because it's service units, right? They're not going to hurt themselves in the process of trying to do an experiment, right? So that's been wonderful. So that is definitely something that will continue as well. Other direction are trying to set up models of ischemic stroke in an in vitro setting. So basically using this as a tissue engineered approach with our hydrogel materials, encapsulating cells, and then providing different cues that emulate things about ischemia in a tissue culture environment. So that's another direction that we're really trying to explore, just getting started with.
Yeah, great summary, Kyle. And it's really exciting to get your work into an animal model, right? It takes so much work to do that. So it's really exciting to finally get there. So congratulations on that. So why don't we move to Dr. Ozdemir? If you could provide a summary of your future work.
Certainly. So I would divide into two main parts. First, using the synthetic materials, we want to basically control the directionality, alignment, and dimension, as well as basically the way we introduce the chemical factors using synthetic scaffolds. But also we are interested in how the ECM assembles along with the scaffold, because a lot of biomaterials cause foreign body reaction and primarily defined by excessive collagen or like deposition, where we believe that not only the physical properties of the biomaterials, but the way the cells assemble their materials matter. That's why we would like to explore this ECM deposition by introducing, for example, hyaluronan-binding peptides to allow the HA to deposit before collagen. And it is known that during embryonic development, in an embryonic wound, HA is the first ECM molecule that is deposited, as opposed to an adult healing that's replaced by collagen. So we would like to study the control of assembly with different peptide sequences like collagen hybridizing peptides or hyaluronan binding peptides. So
yeah, no, I think that's really interesting to see how the cells use the fibers to make their own matrix and how you can change the parameters to have them do different things. So I think there's a lot of, yeah, go ahead.
One more thing to add, of course, is relation to cellular plasticity, EMT, and the fibroblasts, the myofibroblast transition.
Sure, and a lot of people are putting fibers in gels and changing the parameters to see how stem cells change. So there's a lot that you can do with this area of work. So that's exciting. Okay, so we'll finish up by having Dr. Sugimura to talk about his future.
All right.
Go ahead.
Thanks. Yeah, future direction. Yeah, I have two laboratories. So my primary laboratory in Hong Kong, University of Hong Kong, focuses on more fundamental aspect of cancer immunology. So we were trying to understand how immunocells interact with cancer in precisely like single cell technology. We were tracking that trajectory of which kind of immunocells can successfully target cancer, which one cannot. Then actually applying these essence like molecular network in synthetic gene circuit, more like a synthetic biology here. Rewiring like basically macrophages and other innate immune cells that we can easily generate from iPSC. So that's basically one approach, one dimension of my lab, focusing on these fundamental science. The other one, another institution in Hong Kong, more like industry oriented. So that part doing a lot of generation of chimeric anti-gene receptor immunocells like macrophages, working on transformation research. So that's obviously like within 5 years, I would see some products or potential data preclinical model. Yeah, thanks.
Yeah, I really think the idea of, harvesting immune cells and having them interact with cancer, I think that's a really great area of investigation. So thank you for those insights. So again, thank you to all the awardees for their summaries. Before we have it be an open floor discussion or questions and answers, I just want everybody to know that the next submission deadline for the Young Investigator Award initiative is we've moved it to July 31st. So if any of the awardees know of other young investigators that might be eligible, we would really appreciate it if you could share that information with them. Any members of the editorial board, if you'd like to share this information about our initiative, we still have a month to have all the submissions come in. So, so now we'll transition to our open floor discussion. We've had a lot of great comments here, a lot of great and interesting research. So if you have any questions about any of the awardees' research from the editorial board, feel free to ask. Or if any of the awardees have any questions for us, it's basically an open floor. So with that in mind, do we have any comments or questions that somebody would like to start off with?
Yeah, I actually had a question for Tuba. You mentioned that you're functionalizing the materials with HA binding peptides, if I understood that correctly. And so, and that changes something about the fibrotic capsule. So you're like localizing HA, or you're using that binding peptide to bind something else on the other end. We also work with HA and various binding peptides. So I was really curious to hear more about that detail.
Yeah, happy to talk about that, because I also, during my postdoc, I developed HA-based hydrogels. And right now, our strategy to attract the endogenous HA to the surface, because we believe that the molecular weight is an issue. So initially, we started as conjugating the HA binding peptide to the surface so that it can attract the HA, and we know HA organized collagen. So, but then we found that actually HA binding peptide treated surfaces also affects the alpha SMA expression on fibroblasts. So it diminished the alpha SMA expression in addition to increase the HA deposition. So with Alcine blue, we stained these surfaces, and there was significantly more HA, but less alpha SMA by fibroblasts.
Thanks.
Okay, thanks, Kyle. Other questions, comments?
I was very happy to see you, Kyle, asking Tuba something, because I thought I would otherwise would have to prompt you to get in touch, because I would have asked, well, what sort of volume do you inject into which tissues, and would it be a case for basic research and looking at epithelial cells turning their fate into mesenchymal, as you did, to also to then manipulate by using different molecules to manipulate that process? Would that be an idea? But first of all, what sort of volume can you inject, Kyle?
Yeah, so our very preliminary analysis tells us that for a rat brain, which is quite a bit larger than a mouse brain, which I think people are more familiar with, like 12 to 14 microliters is about as much as that we could actually do. But that seems like it might be sufficient, because we don't want to inject too much, because then you create pressure, which has a negative consequence on the presumably healthy brain tissue as well. So, so that's our current plan. Yeah. So.
taking off from Kyle in terms of the amounts that we are currently getting. We initially started with a silica glass to understand the effect of the binding peptide. So at that time, we really didn't quantify how much HA was being deposited. Later on, we created these nanofibrous scaffolds, and with that, we increased the surface area. So with these scaffolds, we are currently trying to quantify, because we have the surface area quantified by each discs that we make. We are using polycaprolactone based nanofibers. So we will. So the final answer is we don't know yet how much HA we are producing per gram of the scaffold, but we are in the process of doing that. And the other part of your question is implication to EMT. So another project, actually a student is working on that part for the keratinocytes. With the large diameter, we know that they behave like more mesenchymal, cells. So we are actually also investigating whether HA binding peptide treated surfaces on the more mesenchymal-like extracellular matrix mimic, can we get at least recover some of these epithelial markers? And we are in the process of developing the fibers for that project too. So we are just getting started, as to say.
Great, thank you. That sounds a good project as well. Thank you.
Yeah, very interesting. I see a lot of overlaps amongst the topics people are working on. And just follow up on EMT as well, if you're interested in connecting to some EMT specialists. I think me, myself, and I think Christoph and also Kat has been working on EMT for quite some time. And we're looking from various aspects of, perspective, cell biology, matrix, as well as disease, possibly, and fibroblasts. And we have some work going on the fibroblast cells as well. So I'm sure there will be, if you feel that you want to get yourself connected with someone working on EMT more specially, or maybe some special cell types of the EMT, I would be happy to discuss. You can always email me or email other people, editorial board members and editors, for suggestions and or possibly for collaborations as well. Yeah.
I will be happy to, and thank you for the support. I will be interested, actually. We are currently limited with some, we are primarily focusing on the fundamental aspect, limited with some cell types, but certainly it would be great to be in touch. And we have a nice imaging facility in my program. We have SHG, supported second harmonic generation microscopy, supported with CARS, which we can get some chemical information in addition to basically non-invasive imaging, so that we can distinguish collagen signatures from hyaluronan signatures. That's what we are hoping to, understand, because with these peptides, cells are depositing the HA first, or, they are just depositing HA more, or, so on and so forth. So I think there is a lot of questions to be answered, and it would be really nice to consult with your expertise and maybe form new collaborations in the future. That would be great.
Yeah.
Okay, great. Other questions, comments?
Well, I would have a question to Sarah, because of the large animal. What sort of large animal model will you use, and what is the weight? What role does the weight play? Because in the human, of course, there's the upright posture that causes most stress. Can you simulate something similar with with large animal models, which are the most popular ones for in your case?
Yeah, thanks for that question, Christoph. We actually get that question all the time. Reviewers love asking this question. Grant reviewers, paper reviewers, we get this all the time. So the model we're using right now is actually the cervical spine of large frame goats. So the goat cervical spine has a couple advantages: that it's semi-upright, so it, not as completely upright like us in bipedal animals, but it is semi-upright. They keep their heads upright for a lot of their life and existence. The sizing is also very similar, so the geometry and mechanical properties are quite comparable to at least the human cervical spine. Nothing is really very similar to the human lumbar spine, unless you're talking like kangaroo or other inaccessible animals. But the goat cervical spine is very similar in dimensions to the human cervical spine. So that's a benefit. With respect to the quadruped versus biped, there's actually been a couple of biomechanical studies by folks in the spine field that have suggested that and proven that actually quadrupeds, because they have to maintain their spine in a horizontal position, it actually requires a lot of activation of the paraspinal muscles to maintain that horizontal spine. So this basically causes axial load across the spine, just as it is in bipeds. So the loading mechanism is still primarily axial, even in quadrupeds along the length of the spine. And there's some evidence that actually that loading is maybe even higher than in humans, as the bone density of animal vertebra is actually much, much higher than it is in our spines. So by Wolff's law, you would assume there's more load perhaps on the spine. So even though we're limited to quadrupeds for a lot of our spine work, we do think they are a pretty similar model to the human condition.
Yeah, that sounds very convincing. Thank you very much. That's, I think
Glad I could convince you.
Yes. Anyway, it's just at the heart of CTO as well, because we like to draw the connection to clinical medicine. And I'd like to underline what Ryan said about the review process. If we hear so often that with our own papers that then we get reviews back to say more research, more experiments, and we at CTO like to think it's more important to choose the right wording to make the connections with ongoing research in other fields, and to have a perspective from the impact of your research towards clinical medicine. So I can only encourage that. What Ryan said about the review process, and well done for your perspective as well. Thank you, Sarah.
Thank you. Yeah, totally agree.
Yes, as Christoph mentioned, the previous incarnation for CTO was Acta Anatomica. So we have some sort of tradition of specializing anatomical analysis, comparative anatomy. So your large animal model would actually be quite suitable, even if it's not related to stem cell research. It will be actually quite interesting for us as well to see this kind of work, right?
Good to know.
We actually, we welcome you to consider CTO if you have actually.
We're doing some studies right now to finish up a set of experiments. So we're probably anticipate submitting a paper somewhere the end of this year, depending on how things go. So these large animals model studies obviously take a lot of time and a lot of money. So it's important. We try to get as much data out of them as we can possible.
Cool.
Okay, so other comments, questions?
I'm wondering, Kat. Sorry, you have some thing to say about the publishing. I think you've been involved in as an author and editor. So I guess for young investigators, oftentimes choosing where to publish is a big thing. Because it's their colleagues' view that they should be worried about, and always maybe the better impact is better normally. But I assume, of course, the quality of science is important, and in the end, where your heart lies is going to be, a place where maybe you can do most contribution, have most contribution of, right? So I guess, do you have any comment about how they should
do have
approach?
comments. So I can tell you my opinion regards both as being on editorial boards of journals, but also as a kind of in a leadership position in a department. What do I tell my junior faculty? So I always feel that one should publish upload preprints, because I think that's very important. And I tell all of my faculty, and it's a mandate in my lab, when we submit to a journal for review, we always upload to bioarchives. I know that many institutions and the NIH will not, they'll take into account a preprint. There will be discussion. They don't count it the same as a paper. But when, I was at study section on Friday, and we were discussing applications where there were preprints. And, in a way, the preprint is good because you can evaluate it for what it is. There is no label attached as to, is it, in an impact factor journal. So I think that's kind of really important. I also think, like Kyle, I've had not that many papers rejected in my career because I also feel that, there was discussion about aiming high. The first, when we submit, we like to aim high. I don't actually feel that's the best approach. I think one has to be realistic and go to colleagues to determine where the work will fit, and then to submit, within reason, rather than, there are labs that will always submit, their first submission will be to a CNS journal. I don't think I've probably submitted one paper to a CNS journal in my career. I think that's kind of, you can have aspiration to go to these high impact journals, but what they ultimately, the problem ultimately is that, if you go there, you'll either get a flat reject, which in a way is a blessing in disguise, or you can get reviewed. If you get reviewed, that may set you back 3, 4, 5, 6 months if you don't get published. And I always tell my junior faculty, yes, you aspire to go to these fancy name brand journals, but when will your paper actually be published? And I think that's really important, rather than tagging it with, well, it could, I wanted to see if it, might it go to Nature? It doesn't matter. Send it to where it really belongs, rather than playing this game. And then you go through all these rounds of reviews, and you could be there for some extended period of time, by which point the work should move forward. And I feel too much are we obsessed with what is the brand, rather than the quality of the science. And I feel that in the end, if a paper is written up and it can be peer reviewed and published, well, as you said, Gojan, it's the quality of the work will be appreciated in the long run, and it will be sort of cited highly. I'm also a big fan of eLife. My lab have published extensively in eLife in the old model. It'll be interesting to see how the new model that was rolled in January will kind of evolve. But I really feel that all these boutique journals, I find it a little bit of a distraction. And I think moving forward, we need to get away from branding and tags and impact factor, because I know in my department, it's not where you publish, it's what you publish. And ultimately, I would argue that a junior investigator with multiple impactful papers in their field that will ultimately get good review letters, because when we look for promotions, it's not grants and papers, it's what your peers, how your peers evaluate you. And I think someone with multiple impactful publications versus one high impact paper will probably do better in the long run. And I think they'll do better at the NIH study section. I expect they will do better for promotion, at least in my experience in the United States. I know that in some countries, you have to, on your CV, put impact factor of journals, citations of the paper. I think this is really a time waste. That's just my opinion.
Yeah, I think that would be a fantastic, idealistic kind of way of looking at things. And I think some universities in the US, and I think especially also in Europe, are actually really pushing towards this kind of, publication and also evaluation systems. But in some rest part of the world, I think, and in some other countries, Hong Kong and Japan is definitely also like this. Yes, because admin has a big role to play in terms of deciding the promotion and grant, and most of the time they don't really care about the content because they can not probably evaluate the content, but rather looking at the impact factor. So I think this is a kind of realistic way of, for facing a lot of PIs.
No, I
guess,
totally agree.
so.
But yeah, and I feel the admin, this is where I feel maybe I'm privileged because I'm in an institution or I write letters for colleagues for promotion where, people will take my opinion, not some number that is calculated, by someone who has no appreciation of the science.
All right.
Yeah, great perspectives. Thanks, Kat. Thanks, Gujun. I think we had some other questions. I think, John, did you have a question?
I did, but it was very unrelated to this.
That's fine. That's
Okay.
fine.
No, I had
a
a question for Sarah. I was interested when you're talking about your tissue engineering approaches. Like, how do you envision, like, if everything works, like, are you like implanting, like removing a vertebrae and putting in a brand new one? Like, what is your vision for this?
Yeah, it is a kind of a lofty vision. It's sort of a crazy, idea. And it's taken, honestly, this isn't just my group. We have a really large collaborative team at Penn that is composed of other biomedical engineers, veterinary surgeons, human orthopedic surgeons. So this really is a team collaborative undertaking. So our vision is that, someone comes into our orthopedic surgery and they have disc degeneration, back pain, they would be a candidate for fusion surgery. But hopefully we would be able to isolate some kind of stem cells from them. Which source of stem cells, that's still up for determination, whether that's adipose, bone marrow, there's a lot of options. iPSCs, you need a lot of cells to make a tissue engineered disc this large enough to put in a human. So that's a big challenge we're encountering as we start moving from animal cells to human cells. So cell source is an important hurdle, but hopefully we could get, something from, some source of stem cells within that person's body that would be able to generate enough cells for us. We then seed these on our scaffold. So we use electrospun PCL for the annulus analog. We use a hydrogel for the nucleus analog. And then we have these end plate interfaces that attach on in culture, which was the focus of the CTO paper.
Any other comments or questions?
Yes, I also wanted to thank Kat for her advice for her young faculty to use the preprint server, because that has got the educational side effect that you get things done, and the threshold to put it into writing or to submit it is quite low, but it's an incentive to get finished with your work and to put pen to paper and get to a point where something is fixed, and nobody will scrutinize this bioarchive, but you've got a date of the primary publication of those findings, and I think that's a very good incentive for young faculty to use preprints, and we should encourage that more and more. It's new to me entirely. It was new to me entirely, of course, but I think it's something we should use always. Thank you, Kat.
Okay, great. Thanks, Christoph. Any other questions, comments? Okay, so if not, I would like to thank Simon for organizing all of this. He's done an excellent job in working with all the young investigators. So thank you, Simon, for all your hard work. Thank you to all the awardees. I worked with all of you on your submissions, excellent submissions. So I really hope that you consider submitting to CTO in the future. So thank you for your submissions, and I wish you nothing but the best in your careers. It looks like many of you are starting off on a great trajectory. So really, really nice to have you here today. And also thanks to several of the editorial board members for attending and offering advice and perspective and questions. So thank you for attending. So again, as a reminder, submissions to CTO can happen at any time. Christoph, Gujun, and myself would be happy to work with you on your submissions And we also have the next installment of the Young Investigator Award. Those submissions are due end of July. So again, talk to your colleagues and try to encourage them to submit to be a part of this initiative. And with that, I think we'll end here today. So thank you again for attending, and wish you all a very good rest of your day. Thank you.
Thanks, everyone. Nice meeting you all.
Yeah.
Thank you.
Thank you, guys.
Thank you.
Thank you. Have a nice day to everyone.